All right, now let's talk about physical barriers of innate immunity. So let me write it down, physical barriers of innate immunity, right? So innate immunity will have mechanical barrier, physical barrier, chemical barrier, and this one will specifically be discussing the physical barrier, and this is the course, innate immunity course, where you will be understanding the non-specific immunity. So just imagine that we have the outer protective layer where microbes, they are trying to enter, but this layer is basically protecting and blocking the entry, right? Blocking the entry inside our body because we have the mechanical physical barrier, which is skin. Okay, so let me write it down. Skin basically protects us from the microbes. And further, you have mucous membranes. Specifically in our body, we have mucous membrane. We have respiratory tract. In respiratory tract, we have various systems available that can block the entry of microbes. And further in the gastro, let me clearly write it down, gastrointestinal tract. In our GI tract, a lot of microbes are already there. So we need a system for that to block those microbes. We have genitourinary tract, basically, right? So that tract is basically, I've written wrong over here. It says, then you have eye defense, which protects our, our body from microbes. So let's talk about skin. Skin has 3 layers, right? So you have outer one, then you have second layer, then you have third layer. The outer one is called epidermis, right? The second one is called dermis. The third one is called hypodermis. So you have outer one, you have middle one, then inner one. Let's talk about the outer one, which is epidermis. Outer epidermis layer, where the protection and fighting happens. So this is the protective layer where the protection happens. Then you have antimicrobial barriers. So AMPs, antimicrobial peptides, these are small molecules, these tiny molecules. They can kill microbes by different mechanism. Then let's talk about keratocytes. Keratocytes are really important cells that play an important role in the immune system. They are the dominant epidermal cells, the most dominant epidermal cells, right? And their function basically is protect the body. It produces the keratin protein. You have tight junctions that are present between these particular keratocytes so that the entry of the pathogen is blocked. Don't have any space in between them. You have antimicrobial molecules. So these green color dots you're seeing, these are AMPs. They help in cytokine signaling also. Now we understood that cytokines are the immunomodulatory molecules, and they are involved in this one also. So they are not just passive cells; they are active immunocompetent cells. They provide protection. Next is let's talk about the important molecule, which is keratin. Keratin protein, keratin polymeric structure, right? So keratin is a polymeric fibrous protein. So it's going to create that mesh-like structure inside our body, and which will be highly challenging to degrade. So it's highly cross-linked. Keratin is basically highly cross-linked fibrous protein. It's like a fibrous protein so that it can trap microbes. So it's basically insoluble protein and highly strong protein. That's the next point. Next point is it's resistant to enzymatic degradation. Enzymes are not able to degrade this particular protein. That's the beauty of this particular protein. So it provides highly protective surface. Okay, if the surface is created by keratin, so highly protective surface will be created, and then basically microbes, they get trapped. Microbes, they can't enter inside that particular barrier. Now let's talk about tight junctions. So we understood that there is a protein complex between the cells, right? So if I draw the cells, these keratocytes, right? In between the space, you can see there will be the protein complexes in green color that I'm trying to show. These protein complexes, they are the tight junctions. They will fix the cells so tightly that nothing will enter in between them. So they will prevent, right, any kind of entry of the microbe. So there will be prevention. Epithelial integrity will be maintained, right? And the proteins that are involved are claudins. You have occludins. These are the proteins. You have JAM, which is junction adhesion molecules. Really important proteins that are involved in tight junction creation. Okay. Now let's talk about another aspect, which is skin microenvironment. Skin also have its own environment which can protect the body And what is that environment? The first one is it provides the unfavorable environment for pathogen, which includes one which is acidic environment. So skin will have acidic environment around 5 to 6, right? 5 to 6 is the pH of the skin. That is not good for microbes. So sebum, sweat, fatty acids, specifically coming from good bacteria that is present on our skin, will kill the microbes or will just stop the growth of these microbes because they don't like acidic environment. So microbes, they don't like acids. So they will be inhibited, they will be killed, right? All right. Now let's move on to the next aspect of our physical barrier. Let's talk about AMPs, antimicrobial peptides. So cells, they produce antimicrobial peptides, specifically keratinocytes. Okay, let's talk about keratinocytes. They will produce defensins. It's also antimicrobial peptide. You have cathelicidins, another antimicrobial peptide. What are these? These are cationic peptides, positively charged peptides. So what they can do is they basically can create holes in bacteria, and because of that, bacteria can get killed, microbes, they can get killed. So they target the membrane of the microbes. If we talk about mechanism of antimicrobial peptide, as first one, it's going to get inserted into the membrane. Once it gets inserted into the membrane, pore formation will happen, right? So next point is pore formation will happen. And because of the pore formation, right, the ions or the molecules that are present inside the cell, leakage will happen, and that will cause their death. So as you can see over here, these yellow colored dots, they are coming out. That will cause the death of the bacteria. Target in this case, bacteria will be targeted. Fungus will also be targeted, as well as viruses, enveloped viruses, they are going to get targeted. Okay. All right. Now let's move on to another term that we use in case of skin immunology, which is skin associated lymphoid tissue, SALT. So it's basically the immune system of skin. So we have various immune structures that are present in skin, and the SALT system is used for surveillance, just to check if there is any kind of foreign invader. It's used for antigen presentation because then further activation of immune system is also required. Then immune activation is important. And what are the components? You have component number 1, which is Langerhans cells. Okay, really important cells of SALT. And then you have intraepidermal lymphocytes. Intraepidermal lymphocytes present in the skin. Then you have dermal macrophages. In the macrophages are present in the dermal layer of the skin. Let's talk about Langerhans cells first. Langerhans cells, these are nothing but dendritic cells. Okay, so let me draw those dendrites, beautiful dendrites. These are the dendrites, and this is their dendritic cell present in the epidermis, and that is why these are the antigen presenting cell of skin. So these are nothing but dendritic cells. They are used for antigen presentation, which is APCs, and then it's present in epidermis. They are used for phagocytosis. Obviously, they will be used for antigen presenting. So antigen presenting cells of the skin, they will migrate to lymph nodes. Lymph node migration should happen because activation of immune cell is required. Next is naive T cell activation. They will activate further the T cells, naive T cells that are mature T cells coming from the primary lymphoid organs. So it's basically nothing but a bridge between innate and adaptive immunity. These Langerhans cells, these are dendritic cells. So you can understand whatever the function of dendritic cell is, it's a function of Langerhans cells. All right, now let's move on to the next one, which is mucous membranes. Mucous membrane is present in various parts of our body. It's present in the lungs. Let me draw the lungs. I hope my drawing is not that perfect, but you can see this is the lung, which is part of the respiratory system, right? Then you have GI, gastrointestinal tract, which is intestine, and I'm trying to create that GI tract over here. Then you have another one, I hope you can guess, is genitourinary tract, where you have the genital system, and then further you have another one, which is the eyes. In eyes, you also have mucous membrane that will protect our body. These are the potential entry points from where the pathogen can enter. That's why we need more protective system. Let's talk about mucins and mucus layers in detail. So you have mucus, which is containing a protein called mucins, right? The mucin is nothing but a glycoprotein. Lot of heavy sugars are there. So glycosylation is very, very strong. So it is heavily, let me write it down, heavily glycosylated. This protein is heavily glycosylated. If I draw the structure, I can draw those sugar moieties on the surface, as you can see over here. And that represents the heavily glycosylation of the protein. These are nothing but sugar moieties, right? So they trap microbes, the mucus layer, mucin protein. They block attachment of the microbes to our surfaces. It also helps in removal of the pathogen from the body. So it's not just static layer. It's a dynamic mechanical, rather I should write now, biochemical barrier to protect ourselves from the infectious agents. Next is lysozyme, very, very important protein molecule, which is antimicrobial enzyme. This is antimicrobial enzyme. So I'm creating the illustration of the protein, and it is present in tear, saliva, and mucus. Okay, so the microbes contains basically a peptidoglycan structure in their cell wall, positive as well as negative. This enzyme is going to degrade that peptidoglycan, specifically the linkage called beta-1,4 glycosidic bond, which is present between two molecules, N-acetyl muramic acid and N-acetyl glucosamine. N-acetyl glucosamine. These molecules, they are bound with 1,4 glycosidic linkage, and because of that, it's going to cause the osmotic lysis in the bacteria, right? All right, now let's move on to the next component of the physical barrier, which is lactoferrin. Lactoferrin is nothing but it's iron sequestering glycoprotein. It takes the iron from the environment, and you know, iron is really important for microbes. So if I draw the illustration of this particular protein, and you can see these dots, these are iron, and this protein is capturing those irons, iron binding protein. So it binds to free iron, and it limits the microbial growth, because iron is really important for the growth of microbes. It limits the microbial growth, right? So it's going to stop the growth of the bacteria. Now, this is called nutritional immunity. Wonderful term, where nutrition is the central aspect to block the growth of bacteria. So it basically blocks the nutrient, and then specifically iron sequestering, that capturing the iron from the environment. And iron is important for bacterial metabolism, enzymatic function. So if you're not providing iron, bacteria is going to have difficult time in, and our pathogen will have difficult time in and taking over the immune system. Next component is lactoperoxidase system. There are so many interesting systems that are present in our body. Lactoferrin, lactoperoxidase system. Lactoferrin we discussed is an enzyme which does the oxidative antimicrobial reactions. And I hope you remember in the last lectures also we discussed about ROI, reactive oxygen intermediates, toxic oxidants that are generated by this particular system, lactoperoxidase system, which targets the bacterial membrane, which targets the DNA of the bacteria, and then it also targets the protein functions, right? That's very important part of the immune system. Now let's talk about MALT, mucosa-associated lymphoid tissue, right? And the main aspect of this is to immune surveillance system to identify if there is pathogen coming, trying to come into our body. So it's immune surveillance system. And you have further GALT in this one. We have BALT, we have nasopharyngeal lymphoid tissue, gut associated lymphoid tissue, bronchial associated lymphoid tissue, nasopharyngeal lymphoid tissue. All these are the mucosa associated lymphoid tissue, right? Now let's talk about GALT, right? GALT. Now in this one, you have Pierce patch, you have tonsils, you have adenoids, you have lymphoid follicles, adenoids, you have lymphoid follicles. These are all the components of the GALT. Okay. Moving further, let's talk about M cells. I don't know whether you have understood the M cells. What are their functions? Why they are called M cells? Let's talk about that. M cells, they are also known as microfold cells. Microfold cells, and their role is basically in the immune system. So epithelial antigen sampling cells, really important cells. What they do is they sample antigens in our body. So they have very, very limited microvilli, They have thin glycocalyx, okay, thin glycocalyx. These are the feature of these cells. Highly endocytic activity. Endocytic means they take the, molecules from the environment, immediately eat them up. Not eat them up, but take them inside. So what they do is from intestine, from intestinal lumen, they're gonna take those components inside and then activate the immune cells. This process is known as transcytosis. Really important for activation of the immune cell. Transcytosis. So M cells, they've been very, very important for activation of the immune system. Okay, now let's move on to the secretory IgA. Secretory IgA is basically nothing but antibody, immunoglobulin A antibody. So secretory IgA means the antibody that is getting secreted in the mucosal surface. So antibody of the mucosal surface is the sIgA, right? It blocks the toxins, the proteins, the viruses. It blocks the bacteria, and they directly bind to these microbes. And it blocks the invasion, specifically invasion or attachment of these microbes inside our body or to the surfaces. So block invasion is very, very important. IgA is work without activation of a very, very strong immune system. Okay, so work without triggering a strong immune response. It just basically blocks the microbes, right? And it's important for various organs, right? Wherever we have mucus. Now let's talk about the next aspect, which is the respiratory defense system. So in the respiratory system, we also have a lot of microbes coming directly from the air. So air filtration is really important. Our body filters the air with various structures. Reflux action of the respiratory system is really important. Phagocytosis happens inside the respiratory system quite often. And also you have those cells in the respiratory system that will have these structures called cilia, and these cilia basically they beat in upward direction, and because of that, anything that is coming inside our lungs will come out, and it will move upward direction towards the pharynx, right? So it will move like this, these cilia, and the components they're going to move outside. So mucus, it's also the mucociliary escalator. It's known as mucociliary escalator, which will remove the pathogens which are entering inside our body. It will prevent the lung colonization. Anything that is trying to colonize lungs will be taken out by this escalator. Okay, now let's talk about alveolar macrophages. Really important component of our innate immunity. Alveolar macrophages, so I'm drawing the macrophage. They are really important in case of lung immunity. Macrophages, their role, they are present in pulmonary alveoli. Their role is phagocytosis, eating the pathogens, cytokine production, so that immune cells they get activated, and inflammation, so that the local area get protection immediately. So this is the component where alveolar macrophages they play an important role. Now let's talk about GI defense. Gastrointestinal defense, the main component is in the stomach. We have a lot of acid, very, very strong acid is present inside our stomach. The pH is around 2 to 3. Highly strong acid is present, highly acidic environment, and because of that, it can denature microbial proteins. They get denatured in the acidic environment, so that if anything we eat accidentally will be neutralized. It can target membranes, it can target toxin, so it will denature any kind of toxin. Inactivation of the toxin will happen. So it's really important that our stomach will have high, low pH, highly acidic environment. Now let's talk about good bacteria also. Our body have a lot of good bacteria, microbes that are present in green color. I'm designing those, and they will basically fight directly with the pathogenic bacteria, and they'll try to stop the growth of these organisms by various mechanisms. They can take the nutrients, they will occupy the space, they will also produce some antimicrobial components that will target these pathogenic organisms. So there are various ways these microbes target the pathogens. Let's talk about Paneth cells. Paneth cells, they are present in our intestine. They are also part of the immune system. Paneth cells, I'm creating an intestine. Okay, so let's imagine this is the intestine, and you have in the intestinal crypts, the cells that are secretory epithelial cells. Secretory epithelial cells are our Paneth cells. They produce defensins, they produce lysozymes, And all these microbes, all these components, they are going to target microbes. So there is always this balance is being created by our intestine of those microbes. Otherwise, they will keep on growing, right? So we don't want too many microbes in our intestine. Let's talk about genitourinary defense system. So urine flow is one of the important example where flushing of our urinary bladder will happen because of the flow of the urine. Then renal medulla is also very, very important in immune defense. Renal medulla has hypertonic environment. High osmolarity is there, hypertonic environment. And hypotonic, whether it is hypertonic, cells really don't like that kind of environment. And if there is any pathogen, that will be killed. So to stop microbes, we have environment, specifically high hypertonic environment in renal medulla. Let's talk about vaginal microbiota. And in vaginal microbiota includes Lactobacillus species. Lactobacillus acidophilus is the bacteria. What it does is it's going to convert glycogen to lactic acid. Because of that, you will have high acidic environment in the vaginal environment. You have low growth because, as we discussed already, acidic pH is not a favorite environment for the microbes. Next is eye defense. Eye defense, our eye produces tears, right? The tears, and these tears basically they wash our eye and protect our eyes from various microbes. So it contains lysozyme, it contains lactoferrin, it also contains secretory immunoglobulin A. So you can imagine it's complete immune soup, which will kill the microbes if they are present. So they will target, let's imagine this is bacteria. They will completely kill the microbes if they are present near to our eyes. Okay, so now if we conclude our physical defense is working on various aspects. The first one is microbial recognition. The physical defense is going to target the microbial recognition. It's going to recognize surveillance system is there. It also induces inflammation, so that there is, if there is anything that is targeting our body, you have cell signaling, you have adaptive immune activation also happens in our the physical. You can imagine so many things are happening, right? And this is the overview of the physical system. We have covered most of the important aspects of physical system, and then in our future videos we'll be covering other important detailed aspects of innate immunity, and then we'll have a complete course on immune system, right? I'm already in the description box. You will get the link for the full course. In that full course, you will get notes, you will get all the data, you will get questions that you can download. Your learning will be really good. You get certificate and all. On YouTube, you can watch those videos, but I recommend if you're watching all those videos, you just complete that particular course, get certified, and you will also have that confidence that, it's more organized on Research Goal website. So please see the description box. I'll get the link for this particular course. And I want you to complete all the courses that I'll also make in the future, so that it's also, helpful for us also. If you support the Research Goal, I hope that this will help your journey in research and your understanding in the immunology in general, right? All right, we'll meet in the next video. Take care, everyone.